All right, as people straggle in, we're going to go ahead and get started. Please feel free. We always feed people for the Digital Engagement Committee meeting. And so there's some hot tea, as well as some pick-me-up snacks and I think there's some drinks back there. Unfortunately, we're not doing a Digital Engagement Committee dinner at this meeting, and my apologies for that. So first things first, we're going to take roll for the meeting. Frank, I'm just going to read out the folks on line here so that we can get that out of the way. We have Javier Retamales, Michael Pishvayan, Tracy Taylor, Lisa Perez, Kara Laubach, Jennifer Morse, Katrina Peterson, Catherine Koralchuk, Stephanie Groff, Mayor Gadikar, Jack Aiello, Indira Castellano, Diana Ruiz, Adam Torres, Heloise Soares, Christine McLaughlin, Mariam Buddha, Al Rutebembemnois, Zainab Aroglu, Patricia Arlaskas. And then there's someone calling by phone at a 307 area code, if you could identify yourself. Or not. All right, and then Frank, do you want to call the rest of the call? Okay. All right, why don't we just go around the room and we can go ahead and just, I see there are no mics at the tables, so just scream exceptionally loudly and announce yourself. And then again, this is an open meeting, so if you're not on the committee officially, that is totally fine. Oh, great. There is actually a mic coming up, so we'll just pass the mics. This is an open meeting as we cover digital engagement. That includes social media, which in and of itself is an open channel. So everyone is invited, and please just let us know you're here. We'll start with Nagla. Just announce your name and yeah.
Nagla Abdel Karim, or Nagla A. Karim.
Krishna Guntur.
Kathy Crew.
Dana Sparks.
And then as Dana has to walk through three things, can everybody move in? Because they put us in this incredible conference room. So
Zach
can everybody just
Schley.
move in?
Chris Kippola.
Okay, John, go ahead.
Jonathan.
Kara Laubach.
Lee Jones.
Aaron McCaig.
Amanda Stampless. Laura S. Feller.
Roberta Albany.
Joel Poynton.
Joel, you can keep the mic up there. We'll go to Jonathan up here.
Jonathan Summers.
Anne Marie Mercurio.
Becky Johnson.
Jenny Mason.
Barbara Segarra.
Allison Rosen. Amy Geschwender. Lauren Frizel.
Patricia Flowers.
Okay. I think that, has everybody had a chance to announce themselves? If not, please come up to Frank after the end of the meeting so that we make sure that people know you were here. I am really honored to open my last Digital Engagement Committee meeting. We are seeking the new chair, and if you're absolutely interested, please come talk to me. But this has been the best SWOG committee for the last 7 years, and it's all because of the work that's been done by you all. And I know we're just going to get more integrated into SWOG as we move forward. And so I'm looking to see what the new chair brings to the position. We do have a tight agenda here. So I'm going to go ahead and bring, we have a presentation about SWOG 2001. And I believe this is going to be an online presentation, correct? Okay. Dr. Bishwayan, if you want to go ahead and we'll put your slides up.
Yeah, hi there.
Hi, how are you? Can you see your slides?
Yes, I can. Thank you
Okay,
so
can we
much.
put that in presentation mode on the, oh, great, perfect, never mind. All right, go ahead.
All right, perfect. Well, thanks for giving us the opportunity to try to present this study and try and see how we can boost up accrual. This study is a randomized phase 2 trial of olaparib and pembrolizumab versus olaparib alone as maintenance therapy for patients with metastatic pancreatic cancer and known germline BRCA1 or BRCA2 mutations. This is essentially the follow-up to the POLO trial that led to the approval of olaparib as a single agent in the maintenance setting. Vincent Chung is the overall PI of the study, and you can see the rest of the group there. It's open through SWOG, the Alliance, as well as ECOG and other cooperative groups. Next slide, please. So just as a bit of brief background, olaparib is now approved for the treatment of germline BRCA-mutated pancreatic cancer for patients who've been on platinum-based therapy and whose disease has not progressed on first-line platinum-based therapy, essentially in the maintenance setting. And this is relevant for about 5 to 7, in some cases, maybe more patients who harbor germline BRCA1 or BRCA2 mutations. And the nature of the study is that patients of the POLO study that led to this approval is that patients had to have received at least 16 weeks, 4 months of platinum-based therapy, and their cancer was either responding or not growing, so stable at least. And that trial was a placebo-controlled phase 3 trial, and it led to an FDA approval of olaparib based on an improvement in the progression-free survival to 7.4 months with olaparib versus 3.8 months in the placebo arm for this patient population. Next slide. So we wanted to take the next step in trying to improve this maintenance therapy regimen. And there certainly has been a lot discussed about the potential for PARP inhibitors to be enhanced by an immune checkpoint inhibitor. And preclinical studies have certainly shown that PARP inhibition enhanced anti-tumor immunity by increasing the infiltration of cytotoxic T cells. One of the hypotheses as to why this might work is that PARP inhibitors, olaparib specifically, but PARP inhibitors in general, can actually increase neoantigen production and activate the STING pathway to activate innate immunity. Next slide. And Dr. Chung's colleague actually was able to demonstrate this preclinically, showing in a mouse model of pancreatic cancer that the addition of pembrolizumab, an anti-PD-L1 inhibitor in the lab and in mice led to an increase in PD-L1 expression and also led to tumor regression. And you see the tumor samples there on your right-hand screen from control over towards the right to the combination of both the checkpoint inhibitor and olaparib in this mouse model. You can see that there was actually significantly slower tumor growth. And in the H&E and Ki-67 IHCs you see there, again, the combination of the two drugs together significantly led to increased cell death as well as significant suppression of cell growth in the Ki-67 model. So that preclinical data, as well as the hypothesis that hopefully an immune checkpoint inhibitor would enhance the response to a PARP inhibitor, we went to the next slide. We went to developing this clinical trial. So next slide. There you go. So again, this is a randomized phase 2 trial of olaparib plus pembro versus olaparib alone. The treatment scheme is, again, like the POLO study, patients with who are on first-line treatment for metastatic pancreatic cancer and had undergone standard of care testing were found to have germline BRCA1 or BRCA2 mutations, that they were harboring these mutations, and that patients were on therapy for at least 4 months of platinum-based treatment, that they would then be randomized to what is now the standard of care olaparib as maintenance therapy versus olaparib plus pembrolizumab. I should note that in the pancreatic cancer world, because of the import and the value of identifying patients whose tumors harbor pathogenic germline mutations, the NCCN and JCO and ESMO and every guideline that exists recommends 100% of pancreatic cancer patients undergo germline testing for the presence of a pathogenic germline mutation. So this really is standard of care that we should all be doing. Even though testing rates fall lower than what we would want, it is standard for all patients to be tested. Next slide. The objectives of our study is similar to the POLO trial. The primary endpoint is progression-free survival. So we want to compare the progression-free survival for patients with germline BRCA1 or BRCA2 mutations who received olaparib pembro versus olaparib alone as maintenance therapy. There are key secondary objectives, of course, safety and tolerability, overall survival, the objective response rate, and then we're also going to be banking tissue and blood samples for further correlative studies. Next slide. The key eligibility criteria, again, mirroring the POLO study, is that patients have to have, of course, pancreatic adenocarcinoma. They have to have a documented germline deleterious or pathogenic or likely pathogenic mutation in BRCA1 or BRCA2 done by a CLIA-certified lab. Patients have to have received at least 16 weeks of first-line platinum-based therapy. One of the recent modifications for the trial would be to allow patients who were started on standard of care therapy with gemcitabine nab-paclitaxel, which is a perfectly appropriate first-line therapy. If patients are started on that regimen and then are found to have the germline BRCA1 or BRCA2 mutation, they're actually allowed to switch over to platinum-based therapy, and then they have to have stayed on that platinum-based therapy for at least 4 months. So that gave us a little bit more flexibility in capturing the roughly 50% of patients who get gem-nab-pac as their first-line treatment. And then these are for patients with ECOG performance status 0 or 1. The key exclusion criteria really have to do with the timing of the treatment, and that we want patients to have to be within a reasonable period of time from when they start chemotherapy and get randomized, and then they actually go on to the olaparib maintenance. And this includes patients who received adjuvant platinum-based therapies such as FOLFIRINOX in the adjuvant setting, that they have to that they're excluded if their progression was more than 6 months from the completion of adjuvant therapy, because then it would be considered sort of new first-line treatments. And of course, any prior treatment with a PARP inhibitor, those patients are excluded as well. Next slide for the statistics. So again, following the POLO trial, where there was a median progression-free survival of a little over 7 months with olaparib, we expected a median progression-free survival of 7 months in this control arm, and we're hoping for a median progression-free survival of about 11.57 months, which is a hazard ratio of about 0.6. And with that kind of a statistical design, we require 78 eligible patients for analysis, and the total sample size of 88 patients allows for 10% ineligibility. We were hoping to accrue this trial over the course of 3 years. Next slide. So a couple of important points about the study again is that olaparib is a standard of care therapy, so it does require that be retained through insurance authorization. So patients who don't have insurance, that could be a problem for them. For the most part, insurance companies, because it is FDA approved, don't balk at giving us olaparib in this setting. But you can never predict what's going to happen there. I mentioned the opening of the trial to patients who were started on gem-nab-paclitaxel therapy for 1 month, and then they have to be on the platinum-based therapy for at least 4 months. And then finally, we are going to be collecting Streck tubes for DNA and RNA ultimately. And if the patient agrees to, we also would like to be able to collect tumor tissue for subsequent correlative studies. I think that's our last slide, and I'd be happy to take any questions.
Great. So, Mike, maybe you can just start off by saying, where are you with accrual? And what are the things that you're hoping digital engagement can help you address?
So as of the last report, if I remember correctly, we've accrued 25 patients, which is not quite nearly where we want to be in terms of the pace of accrual. When you do the math and run the numbers, there's 60,000 pancreas cancer patients a year, probably 80% of whom have advanced disease. And so we really should be seeing, or nationally in the United States, we should be seeing upwards of several thousand germline BRCA1 or BRCA2 mutated pancreatic cancer patients per year. We know that about half of all, or more than half of all pancreas cancer patients don't get any treatment at all, which is sad in and of itself. But certainly for the 3,000 to 4,000 other patients who are getting started on frontline therapy for metastatic disease. It's really just a matter of testing them. This trial is open across the country. It's open at more than 200 sites now across the country. And so I think the biggest barriers are really just awareness and testing. The national averages for testing, germline testing for patients with pancreatic cancer, unfortunately, even though it's supposed to be 100% of patients, really falls in the 30 to 40% range, which is woefully low. So I think in terms of digital engagement, one of the things is just education to physicians and patients to be considering and asking about germline and, for that matter, somatic tumor testing if they've been diagnosed with pancreatic cancer.
Do you happen to know if there have been issues in eligibility in terms of getting the drug? Because I know you mentioned about insurance typically covering the agent. I'm wondering about a population without.
To my knowledge, I have not heard anything about difficulty in getting access to the drug. So no, not really. I don't think that's been a barrier. There's a handful of patients who have autoimmune conditions that make them ineligible for the study because of the pembrolizumab. But again, that's a relatively small population. My impression is the biggest barrier has just been finding the patients, meaning testing them.
So I think that's interesting. I'm going to open it up to the committee. I know there's a couple of mics going, circulating out. So I think the question for the study team that we can help them with is essentially, can we move the dial in terms of genetic testing? If only 40% of these people are being tested at all, that severely is going to limit inclusion criterion. We're not going to redesign the study clearly. We're not going to, this is not the forum that we have for DE to look at modifications to inclusion, exclusion. So I'm just going to ask for any suggestions because they have come to us to build a social media toolkit or even a social media strategy to help move the dial. And I'm wondering if people have any thoughts about where we could go and help them. Do we have a rep from, no, we do. No, don't we? Do we have a rep from, oh, Becky, yes, please.
Yeah, I have a question. Is there any social media strategy yet? How are they advertising the trial right now?
Mike, do you want to take that?
I don't know that I know of any specific social media strategy for the trial other than just general what's advertised through the websites for SWOG and Alliance and clinicaltrials.gov.
OncLive. So Dr. Karim had mentioned whether or not have we worked with any of our trade or trade publications about this as well? OncLive was mentioned as a potential partner for you.
Yeah, not to my knowledge, but that certainly would be a good idea.
So and any other thoughts from the group as to if we were to do a social media strategy? I think maybe the genetic testing might be one place. Any other thoughts about where we could concentrate efforts to help them?
Could you do it on, could you do it on some of the like Ashkenazi Jewish websites, given that the BRCA mutations are common in that population?
I heard someone else was going to talk on this.
I just wrote in the chat. This is Eloisa Suarez.
Okay. And also, if anybody on the webinar has any suggestions, just raise your hand and I will do my best to make sure you are acknowledged as well. So I think, oh, Nagla, yes.
Just one of the problems, even for lung cancer, for all. So I think we can pair this actually with the need to test.
Yeah, so I'm, thinking forward, just going from what Becky was saying, there are certain BRCA groups that are advocates. They're usually in the breast ovary space, but I think they could be utilized in this way to just help you push for genetic testing. The one that comes to mind is SHARE. Force is the other one. So there's a couple of ones in the, really, they're in the breast and GYN space, but certainly just if we could partner with them to get awareness of BRCA testing and pancreatic cancer up would be probably one of the things digital engagement could help you with. But the other is, what do you think about a social media strategy where we focus in on increasing the testing aspects? Do you think that would be helpful?
Yeah, and Dr. Suarez mentioned a partnership with PanCan, and they certainly are very invested in this concept of enhancing germline testing and genetic testing for pancreatic cancer patients. I think they'd be very supportive of pushing any social media out through their contacts, and there are other social media groups as well. I don't know what to be honest, I don't know what the rules are for pushing out clinical trial considerations, but there are several avenues on social media that we could consider pushing that through.
Yeah, so I think Frank might be able to speak towards the regulatory approvals that need to be acquired for a social media toolkit and what is not under that purview.
Essentially, at this point, anything we're putting out, we run by CRB, even if it's directed to providers as opposed to patients.
Yeah, so why don't we do this as an action item for you guys? Is there anyone in the community who is working with Force or Share? No. You do, Barbara? Barbara, would you mind being our point person for Force or Share and working with the group? So we can certainly help you make inroads in those groups. Jonathan. Hmm. Mm-hmm. Okay. Okay. Yeah, so Michael, there was just a question about a group that might be available for you to get some subsidized genetic testing as part of your trial.
Yes, there is an organization through Emory University. It's called jscreen.org. As we know, BRCA is very common in Ashkenazi, but it's open for everyone. I believe it was created by the gentleman who started Home Depot, and it works with your GP. And the geneticist there will review your results and send it to your doctor.
That's great. I think you mentioned already, Michael, but Heloise Soares was asking about, is there a partnership with your investigators and PanCan? And more broadly, is there a relationship with PanCan and SWOG? But can you answer the first one more so that we're clear?
Yeah, I mean, I personally have a very close relationship with PanCan and Let's Win PC and Let's Garden. So most of the leading pancreatic cancer advocacy groups. And I think that they would be very supportive of pushing this out on their platforms. It's just a question of what we're allowed to do from a trial point of view. And if we just need to write something for central IRB approval, then that would be fine.
Yeah, I think I would probably err on the side of getting central IRB approval because even if it's a research synopsis, we can get that approved and then they can use it on their websites because it does count as advertising.
Yeah, makes sense.
Yeah. And then the final thing is, there's a bunch of ongoing conversations with industry about this whole idea of a digital opinion leader in oncology and what to do with them and how to utilize them in this way. There are several in the pancreatic world who I would consider as Twitter DOLs and who are friends of SWOG. The one that comes to mind the most commonly is Mark Lewis. So I think if we could get Mark engaged with your team as sort of a social media lead, I think he can reach his platform is actually incredibly active. And I think if we can find a couple more, Joe McCullum comes to mind as well, and he frequently cites Pan CSM, the pancreatic social media hashtag. I think if we give them the toolkit and ask them to participate, I think it might get more eyes on your study.
I like it.
Yes, Allison Rosen.
I'm a patient advocate on a few different studies where I'm go into some of the groups that are online on Facebook that are cancer specific or whatnot specific, and most of them have IRB approval and there's specific social media posts. And so me as a patient and survivor posting in there also has gotten more people to enroll in some of the trials I'm a part of. I think definitely you need providers, you need to work with these community organizations, but within the community organizations, see if there might be a patient advocate that might be able to go on Facebook, on Instagram or whatever.
Yeah.
And again, it's all IRB approved, but so I'm able to go in there and I'm sort of one of them. I'm trusted, they know me, and so they've gotten increase in accrual because, you know, investigators can't go into these private Facebook groups or whatnot. But I mean, I'm sure Mark Lewis knows some other, you know, patients and survivors that could also go into some of these smaller groups that might be on Facebook. So
I think that's a great idea. Yeah, I mean, if you can find a couple of folks, even that you work with yourself personally, who is engaged in these online patient communities, I think that might be another worthwhile thing for us to put our toolkit in front of them, and also the major talking points. And I think Frank and Barbara and I can certainly help put that together as an action item for you.
That sounds great.
Okay, so let's move on. We have study S19 2022. The conversation is going to be similar to what we just had, but again, just we wanted to hear from the investigators about where they would like us to help them think about. And who is presenting this?
I am.
Oh,
Kate
Dr. Peterson.
Yeah, I'm Kate Peterson. I'm at WashU in St. Louis.
Okay, we'll just get your slides up. Yep, go ahead.
And
Welcome.
thank you very much. I was kind of drafted into this last night. Dr. Overman sends his regards from somewhere over the Pacific right now and is sorry he couldn't make it. So I'm pinch hitting. He didn't apparently share with me the fancy slides, so it'll be pretty brief. But I am the Alliance lead of SWOG 1922, which is a randomized phase 2 selection study of Ramtaxol versus FOLFIRI for patients who have previously progressed on at least one line of chemotherapy and who have a small bowel adenocarcinoma. So this was actually activated at the best possible time in December of 2019. And didn't enroll its first patient until June because obviously COVID hit in the months in between. And it is, you know, at least site approved at about across NCTN, 459 sites, and enrollment's currently at 24. And we'll delve into a few of those numbers here in a moment. If you could go to the next slide. So the impetus for this trial is the fact that small bowel adenocarcinoma has precious little prospective clinical data to support how to treat these patients. And what we're increasingly realizing, including, you know, for instance, through studies of comparative genomics, in terms of a longstanding recognition that patients with small bowel adenocarcinomas really underperform in terms of outcomes when you compare it with colorectal cancer, which is what's been, you know, mostly extrapolated to treat these tumors. But from what we're seeing is that while yes, you know, there are some common players with colorectal and also with upper GI malignancies, you see a lot less driving mutations through the beta-catenin pathway. KRAS is mutated at a roughly equal rate to colorectal cancer. BRAF mutations occur a little bit more frequently, however, are only very, very rarely V600E driven. You do see at least twice the amount of MSI high as well as TMB high malignancies as well in small bowel adenocarcinomas. The real big problem with them is that they're very rare, so that makes them hard to study. Traditionally, they've been the most common histologic subtype of cancer diagnosed within the small intestine, although in recent years, neuroendocrine has taken that over by just a little bit. So we see about 3,500 cases diagnosed each year here in the US, about a similar rate throughout, a similar overall incidence across Europe. And there doesn't seem to be a huge amount of geographic variation, but again, studies are relatively few and far between. And you can go to the next slide. So there have been, to date, 7 prospective clinical trials completed for small bowel adenocarcinoma for the entire disease. All of these have been completed in the metastatic setting. And of those 7 trials, 5 of them were for frontline therapies, all of which were oxaliplatin based. So either Capox, Folfox. There was an NCCTG study several years ago of 33 patients to Capirinox, so basically capecitabines instead of 5-FU and FOLFIRINOX. In the second line and later setting, there have been only 2 studies that have been published to date. This one was a trial run at MD Anderson led by Dr. Overman, which was a phase 2 study that was originally meant to look at whether taxanes might be active in a molecular subtype of colorectal cancers, those that are CIMP high. However, they did add on a refractory small bowel adenocarcinoma cohort due to some of the genomic differences that they were seeing, genomic and epigenomic. And so they gave single agent Abraxane basically in 3 weekly intervals. It took them 2 years to enroll 25 patients overall. 10 of those patients were small bowel adenocarcinomas who had anywhere between 2 and I believe 5 or 6 prior lines of treatment. And the colon cancers, they saw maybe a couple of patients who had stable disease, but overall most had progressed, so no responses whatsoever. However, in the small bowel cohort, they had a response rate of 20%, again, very small. Disease control rate was 50%. So certainly that was interesting as a signal finding study because, again, taxane's not active historically or even in molecular subtypes of colorectal cancer. However, we increasingly recognize that it might have utility within small bowel adenocarcinoma. So very small cohort, but an interesting signal. The only other trial in the second line and later setting enrolled a total of 40 patients. That was the ZEBRA trial of single agent pembrolizumab. It was in all comers. So I will say that that 40 patient trial is the largest trial that has literally ever been completed for small bowel adeno. There is no single prospective data set taking a look at FOLFIRI in small bowel adenocarcinoma. There have been no comparative trials that have been completed to date. There's only one randomized trial prior to the SWOG one that's been opened. That one is underway still. It's in the adjuvant setting, so I'm not going to talk about it much, but obviously it's a wide open field with not too much competition and a lot of unmet need. So next slide, please. So we are randomizing patients who have had at least one prior line of therapy, ideally oxaliplatin based, though that is not a hard requirement. ECOG 0 to 1, very, the main exclusionary factors are basically if they have had bowel obstruction, perforation, or have any contraindication to receiving a VEGF inhibitor like ramucirumab or any of the other drugs. It is randomizing 1-to-1 Ramtaxol versus FOLFIRI. So again, not maybe the most sexy regimens, but certainly very, very important in terms of better understanding this disease and seeing how actually, we can impact its treatment. So while this opened, as I said, in December of 2019, with the first patient enrolled in June of 2020, we have to date enrolled 24 patients. That's 28% of our accrual goal, which is 84 patients, 42 patients per arm. So we're at about 28% overall. This is at a rate of about 1 patient per month. Now, to compare it with some of the other trials that have been done, the NCCTG study of Capirinox was performed in 33 patients, and they had it open at 15 sites, and it took them 6 years to complete that trial. That averaged out to about 0.45 patients enrolled per month over 73 months that it was open. The Abraxane single institution trial that Dr. Overman conducted, they enrolled their 10 patients over 24 months at just that one site. And again, pretty much the same accrual rate as that cooperative group study, 0.42 patients per month. And Zebra, again, the largest study and most comparative to this population that we're looking at, we enrolled 40 patients there over 15 months, and we're accruing 2.7 patients per month on that trial. So I think that in some way set our expectation maybe a little higher than we should have, because there's a lot more enthusiasm among patients to receive immunotherapy compared to chemotherapy, especially if there's any travel involved. It's definitely seems less exciting, and a lot of I think folks have overlooked this pivotal trial.
And is that Zebra going for the same population?
Yes. Yeah, it's done. It's completely accrued, so it's not even a competing trial at this point. That was enrolled from 2017 to 2019.
So, okay, one question for you, Dr. Peterson. Is there an accepted standard of care in the absence of data for this population?
Yes, we've created NCCN guidelines, which is, taking the shreds of prospective data that we have, which as well as having by necessity to do some extrapolation or pulling from what we've seen in retrospective data sets. So the big difference is there, for instance, from colorectal, is that frontline, we do tend to favor oxaliplatin-based regimens. It doesn't mean that's necessarily better in small bowel adeno, but we just literally, again, have no data prospectively with the Reno. It does not recommend EGFR inhibitors because there's been no signs of activity in the very, very small studies that have been attempted, even in RAS wild type patients. Lonsurf and Stivarga, and I'm sorry, I'm using all brand names. I realize I'm not really to do that, but they're not even included on the guidelines because there have been really no reports to date on that when we've done literature searches.
Okay, so I'm just gonna stop you there, Dr. Peterson, and ask just a couple of questions so we can move this along. But to Becky Johnson's question for the former, what have you done so far to help publicize or promote this study using social media or other online channels?
So I think I am the only person who's done any promotion on it. I've created a Facebook group kind of couched under the International Rare Cancer Initiative for small bowel adenocarcinoma, which we're all a member of, and like Mike and I are, and some others. Created a Facebook group. It is global, and there are to date 278 people in that group. So I've done some publicizing there. On Twitter, I've posted there. There have been no other tweets or Facebook postings of it anywhere. I can say for a fact, just from people who have reached out to me through that Facebook group, that since I'm the admin there, that we're engaging regarding S1922, we were able to ultimately enroll 3 patients directly from that line. So not a huge, proportion of the patients, but we have gotten a few from that.
I think that's fascinating. So in terms of what DE can do for you, I'm going to open up to my committee and see if they have any comments.
have the mic real quick. I'll just say, sort of similar to what I said in the last one, there are rare cancer patient advocates out there that are active on Twitter for sure. I know, Rain's not remembering the guy that I know from Twitter, but then within the colorectal cancer space, I can guarantee you as a survivor that some of this rare cancer, they might be involved with some of the online colorectal cancer groups. There's one called Colon Town. Literally, there probably is a neighborhood for this specific type, of cancer that if given the information, I can go in and post it and you might get 3 more or 4 more or 10, I think I'm happy to help. And I know a lot of colorectal cancer advocates that are even more in tune with clinical trials that would post it for you.
That would be amazing, yeah.
Dr. Johnson?
I had a clarifying question about the intersection between SWOG members officially posting on social media versus people doing it kind of unofficially or for their own trial. I mean, it seems like a fantastic workaround to all the regulatory aspects to send it through advocates to post it personally on their site. Is that legit? Because if so, that probably is a very powerful way to do it.
So if you're just, I want Frank's take on this. My take is if you're just going to clinicaltrials.org and saying this trial is open, small bowel adenocarcinoma, more information, go here, and this is the number, and they click, that's fine. You want anything more than that? Right now, we're just taking the viewpoint that review is critical. Frank?
I've consulted with the NCI's Office of Communications and Public Liaison on this, and they encouraged advocates who post like that just to be sure to disclose any connections they have with SWOG, for example. But they didn't specifically recommend, additional IRB review.
I'm thinking one of the things that might help Dr. Peterson, we could certainly do a toolkit, and we're going to hear a bit more about it if you want to hang tight, which then can go to all of the NCTN investigators or PIs who are on social media to utilize. I think Allison is spot on. If we can utilize the online communities in this rare cancer, we can see what kind of catchment that is. But the other thing that might really help with this study, and I would love for you guys to think about it as a trial group, we could actually test tweetorials with this. So it would be a series of 7 tweets, one that builds on top of the other, and you could imagine that the tweet one is like, there's a trial in small bowel adenocarcinoma, it's critical, and here's why, and then you explain in 5 or so tweets why this is a critical study, and then your final tweet could be just reiterate and send people for more information. Would that be something that you would want to work on with DE?
Yeah, absolutely.
Because you're already on social media.
I've been trying to build a following specifically to support small bowel research. So,
well, I think there are certainly some things that we can pilot with a tutorial. I'll speak with Frank, and maybe we'll get two of you together and try to come up with how this might work. Is there anybody else who would be interested in sort of helping with that? Allison, great. And then in the back. Yeah. Oh, no, you can help. It's fine. So Laura is going to help as well. Okay, we have a little group for you. How about that?
I appreciate this so much. Thank you.
All right, sounds good. So this is a nice segue for an update on social media toolkits.
Thank you. Can you bring up my slide set? Okay, let's go back. There we go, okay. Just a brief update on our social media toolkits and use of them. So, it's a set of messages with associated graphics. And we're primarily on Twitter at this point, but we are diversifying. So we put together 10 of these at a time, 5 to inform prospective patients and patient advocates. We try to get these in front of patient advocate and advocate organizations as well. And one of the key goals with those are to inform them about the trial and to direct them to where they can learn more. And where they can learn more is either from our patient-friendly summaries, our plain language summaries, which Amanda does a great job on, and the cancer information service that the NCI runs. So 1-800-4-CANCER is the other source. And then the other 5 posts are directed to cancer researchers and research staff. And for these, we provide an overview of the trial, but we've also been trying to include, especially for trials that are already underway where there are accrual difficulties, targeted messages to address those difficulties. So for example, for S1900E, which is a lung map trial, and it's testing sotorasib in non-small cell lung cancer. Sotorasib was approved by the FDA for non-small cell lung cancer with this particular mutation. And since then, accrual has really dropped off. But the focus of this trial is seeing how it's working when co-mutations are present. And so the messaging that we're trying to get out, or going to be trying to get out, these have just been CIRB approved, really focuses on why this trial is still important, even though sotorasib has been FDA approved. So we're trying to send out a set of messages related to that. And this, I think, is something we will try to do more of, in addition to sort of giving overviews of trials and introducing new trials to researchers. This is one of the graphics that we're going to be sending out shortly. So I'll jump back to that with the PI of the trial. These are some of the others we've sent out recently. We've started trying to include at least one shot of a schema. And so of these 4, 2 of them are patient-directed, 2 of them are provider-directed. And we direct them to the online version of the plain language summary of the trial. This is a shot of one of those. And I just wanted to point out something that's new since the last time we discussed these at the group meeting, which is that there is now a devoted patient section of the website where all of these are available. So there's a new menu up there if you haven't seen it, in the top bar, all the way over to the right, where you can find a list of what trials had patient summaries. And of course, the printed versions are available as well. In fact, some of these are out on the table. Pick them up on the way out. So right now we've got 8 toolkits out and a bunch more sort of in the wings. And so the first week, of course, is to drive prospects to patient summary. The second week, to put these at the top of the mind of providers. So this means that at this point, we've sent out sort of 16 weeks of daily tweets. And so what have we done? Sorry, I can't see those numbers. Just some quick stats. Oh, thank you. So about 180,000 total impressions, almost 3,000 engagements. Engagement being a click on a link, a retweet, a reply, a follow, or a like. And key, almost 500 clicks on the embedded URLs, and those are about half and half between our patient-directed and provider-directed. I've wondered about the delivery approach that we're taking with a 2-week blitz. And so I just did some quick graphs, and these are sort of cumulative by day of the week. Obviously, early in the week, we get a lot of impressions, a lot of engagement, some clicks throughs, and then they drop off later in the week. And then if we look at the 10-day blitz over 2 weeks, sort of parallel lines over both weeks. So I took a look at the, we also, after the 2 weeks, sort of at regular intervals, send out single posts of these as well. And I took a look, and so during those first 2 weeks, we get about 2,300 impressions per post on average. Looking at subsequent posts, they get about the same. So I guess there's some benefit to sort of overwhelming people with these initially, keep putting them in front of people so that they get the message. So I don't see necessarily a reason to change that initial approach at this point. But what we do want to do is go beyond Twitter. And so Facebook is where the patients are, it seems. A quick search on papillary renal cell carcinoma on Facebook turns up a bunch of patient groups, some private, some public. But some of these groups have thousands of members. So many of you are already well aware of all of this. So we have returned to Facebook. A couple of years ago, we had a private Facebook group that was sort of tested out for something less than a year and then closed down for various reasons. So this is just a page for an organization that will allow us to also post our social media toolkit postings on Facebook so they can be shared that way as well. So the takeaway message, when you see these go by, please amplify. We send these out to study team leaders when we initially roll out the toolkit so that they can disseminate the resources as well. They can use their own accounts. These are also all available on the SWOG website, all available on CTSU. Core clinical trials page on the SWOG site. There's sections down below for both patient study materials and for other study materials, including the social media toolkits. All of the graphics are zipped together as separate JPEGs to make it easy to make use of those. And these are also, again, available via CTSU as well. So the message I want to leave with is just when you see them in your feed, send them on. Questions?
Well, I think we just have the beginnings of a very nice paper on social media toolkit. So that can probably be something I will hand off to the next DE chair to help Frank just publicize this work, because I think it's relatively groundbreaking. So Lucy has a question in the back, and I'm going to want Krishna just to just give her impression of how it's been utilizing the social media toolkits in her own study. So go ahead, Lucy.
So I just actually had a question. I'm assuming that with all of the issues with regulatory and all, that we're not able to do like, paid social or anything like that. Is that correct? So just kind of amplify these messages on social.
I'm not quite sure about paid social. We generally have had a very limited budget, and so haven't considered that, but that's something that we can look into.
No, I think you can actually. I agree with Frank. I don't think the limitation is not regulatory.
Okay.
I think the limitation is resources.
Understandable. Thanks.
Barbara.
Is any of this being necessary if they're recruiting Spanish-speaking person done in Spanish?
It is not, and that's a very good question. Amanda and I put together a grant over the summer and submitted it to the Hope Foundation as part of their open call for proposals to fund Spanish language translations of all plain language summaries going forward. Did not include the social media toolkits in there primarily because we don't have someone who is fluent in Spanish to sort of manage that. And so, you know, I could send out initial posts, but I can't engage, you know, in sort of two-way dialogue when people respond to those. So if you have alternatives that would allow us to sort of reach those people, you know, non-English speakers, would be.
And I think, you know,
important
in
to
terms of our
discuss.
strategy at SWOG, just the bidirectionality is so key. As I think unless you exchange in a conversation, you're basically just throwing things out to the wind and walking away.
Mhm.
Yep. Yep.
So that is not Facebook is more reachable than sort of even if
Well, you know, we've actually heard, Barbara, that I have heard in the Rhode Island community that there is a segregation of spaces where communities meet. You know, I think it might be a really nice thing to bring to PAC to survey patient communities and see if we can get some harder data on that, because I think it's important. Nagla? So I just want to thank Frank for all the work he's doing on this. I think it's incredible, and he's really moved it down. And then just to close that out, Christian, I don't know if you want to give your 2 cents on what you've been gleaming off of the social media toolkit. Can someone give her a mic?
Definitely, social media toolkit helped in many ways. With the study, this is the first one that we delivered. So one is, what is the language that we could use? So because that's always the question. So it's CIRB approved, and that was helpful. So used it in multiple venues, mostly on Twitter. So now that's something like maybe we should have used Facebook too. To promote the study, that would have been much more, yeah, so we can still do that. So that was very helpful. One thing, what we cannot qualitatively look at is like how many people are going and retweeting and how many of those are opening the study or enrolling the study. So that's something, I think it's a very difficult quality metric to look at. And it may not be easy to look at. So if we have that, then it also shows the impact of the social media toolkit that we're putting so much energy and support into, and how much it is delivering to. Thank you.
Like, I wonder, I mean, I have no idea, but can you ask in, you know, maybe there can be a trial that asks, how did you find out about this? Like, I know, I tell my friends, well, if you go to this trial, let them know that I sent you, just because I want people to know that when I am tweeting that it actually is potentially working. But I mean, I think that would be, if it's not being asked, like, could there be some, you know, study or some trial that would ask that question to test all the work that is being done? Because I think it's great. I love when I'm being tagged on Twitter for all the SWOG stuff, because I share it, and then other people that I know take it. They do share it again in Facebook groups because they find out about it through me. I find out about it through other people. So I mean, I know it's working. At least it's reaching people. But to to measure that, if there's a way to ask people when they're, you're having the initial interview with them, how did you find out about this?
Yeah, so I think that might have to be at the site level, right? Yeah, so maybe something to think about in ops. Dana, I don't know if you have any immediate thoughts about, where did you hear about that study and when that could be asked of people during the consent or startup process? Or not startup, but when they first start the study? I see. Yep, I agree with that, yep. Yeah, almost like part of the registration form. I think it's a very important point, especially if we believe that toolkits provide value. Becky? Mm-hmm. The only caution I would say with that, because I agree with you, is that getting people to consent to studies is a very complicated process. So if we did not see a correlation between toolkits and enrollments, I would not want to conclude they don't provide value. Right? Okay. Thank you. Very rich discussion. Thank you, Frank, for all of your work. We're going to move to Nagla, who's going to give us an update on an ongoing conversation she's having with the Thoracic Oncology Committee about moving forward with digital health concepts. Oh, I guess we're gonna do, what do you want to do?
No, it's fine with me. I just don't.
I'll just leave it here.
Okay. Let me just sit down here.
We're gonna bring up Dr. Nagla Karim's slides now, not Dr. Tami Mori, just so that we're clear.
It's fine.
Okay, that's fine.
So, hi, everybody. I think, we've been discussing about that concept for quite a while, and it's all, related to the conduct of the clinical trials. Do you wanna, I can sort of like say something about it a little bit. So how many of you have heard this presentation before? Not this one.
This is not the presentation?
Sure. So basically, the thing is, in 2022, coming to 2023, we are still using the same, I would say, the same techniques of our, study coordinators coming with, checking the eligibility paper-wise, and it's not really utilizing all the technology and the potentials that we have. Yeah, so it's here already. So I think I can see it from here, yeah. So basically, improving the conduct of clinical trials is, one of the challenges that we really have to work on, because basically we're still using the old techniques in a time that we really need to utilize the resources that we have. So next. The use of smart forms, which is the, sort of like group of smart phrases that we are used to in Epic or Cerner or our technologies. Basically, they drag the information into our charts, but also drag into our attention. What type of patients are we enrolling in those studies? So basically, this will decrease the enrollment of ineligible patients, and that's our hypothesis, that we want to use this technology to improve our studies. So I want to say that I have S1929, which basically has been planned to enroll 94 patients into step 2 randomization. And this goal was determined because it's taking into considerations that 10% of the patients would have ineligible criteria. So there's 10% ineligible rate for enrollment into clinical studies. So we need to facilitate the screening of eligible patients and decrease the ineligible enrollment rate. That study is almost like finished. So, but basically, I'm just saying that, you know, it's one thing that we need to work on. So there's an extreme need to develop personalized smart form, which, as I mentioned, group of smart phrases to capture the eligibility of each clinical trial. And this should eliminate deviations and implement safety. It can be applied to one prospective clinical trial to assess if this model has less deviations, improving enrollment, meeting eligibility criteria, safety, and also utilizing, of course, you know, resources that we already have. So digital technology basically is of great value in safety, and basically it is a pairing tool to use. Harnessing the ability for those digital tools to collect the data is for sure a way to improve the detection of infrequent events. So from the literature, basically there's also a lot of, you know, so there's like few violations that, you know, can happen genuinely, maybe affecting integrity or safety. There's some potential deviations, they don't have such impact. And sometimes we have a lot of criteria that are not that, I would say they're like minor and so on, and they can be missed, and they can basically get the patient out of a study with a very, very valuable information that we're not able to use. So that's why improving the utilization of the EMR, electronic medical records capabilities, should be attempted to eliminate any of those deviations. Of course, deviations can happen, but we do want to decrease them. So this can be applied in a prospective clinical trial, and pairing this technique with the clinical trial. So again, I mean, I'm just saying that, you know, it's just something that we have to drag this information like vital signs, lab findings, radiologic results, pathology, into the you know, sort of like our documentation and our looking into the patient's criteria, and to determine whether using specific smart form reduces those protocol, you know, violations. So the study design will be that we will want to randomize the patients into arm A or B, and A would be the control arm, and B would be to have the standard clinical trial conduct. In the digital intervention arm, the digital tool would be the smart form, and that's pretty much a schema. And the eligibility, basically, it's really using the smart form in patients in one of the selected clinical trials, having a consent to go as well as the consent of the original clinical trial is something that, of course, we'll be discussing. So these are the methodologies. Is we are going to work on group of smart phrases to facilitate the screening of the eligible patients, and use them within one of our SWOG lung cancer committee trials. It's still under preparation. The proposed question, of course, is that we still need to learn from the literature. It's very challenging because the literature does not really focus on those conduct questions. It focuses more about the findings. So we still need to consider maybe having objectives like to demonstrate, you know, the efficiency of the process. So effectiveness, but also efficiency of the process. Thank you so much.
So again, I want to just congratulate or really send out appreciations to the Thoracic Committee, who's really embraced this idea of how can we utilize digital health tools to make things better. I wonder if there's any comments from the group about this concept. Dana.
Yeah, I think this is an excellent question because basically and not only one side, but a lot of times it's even every investigator or every oncologist, they have their own personalized smart phrases. But what we were looking for is if we unify those smart forms, so not to miss. Sometimes the criteria really, when I'm looking into a patient to be eligible, I'm really looking into the most critical ones, of course, the pathology, this, that. And sometimes there are, and of course, everything is valuable, but sometimes there could be a minor criteria of, some lab finding or a urine analysis that comes later on. So basically, what I'm thinking is if we develop the protocol and develop this as well, so it is easier for the sites to drag these pieces of information into the upfront screening of the patients. Even this for auditing would be so easy because they'll just look and you have all the information there, and I mean there's nothing missing, or even if there's something that would highlight there, then you will notice it up front. So instead of later on, oh, this patient did not meet this specific criteria.
So Dana was just mentioning for the group online that there is a pan-NCI initiative to sort of bring in more standardized templates across NCI for clinical trial builds. And so the proposal would be to utilize that process to assess this question, which I think is a great one.
I think this is a great idea.
So Dana, maybe what I'll do is have Nagla meet with you before thoracic committee meets, and we can see. That's great. Thank you very much. All right, so now we have another presentation. This is just an updated analysis on a SWOG-COG study, and it's going to be presented by Dr. Mordler online.
Hi, everyone. Thank you. Sorry that I'm not there in person. I'm going to share my screen. Can you hear me okay?
Yeah, Scott, we can hear you great.
Oh, it says it's disabled for screen sharing.
Okay. Do I need to do that from my end? Let me see. Oh, we have your slides, Scott. Do you want us to just play them?
Sure.
Okay, let's do that. Do we have Dr. Mordler's slides?
I think you have them done. I don't know if Frank has them.
Oh, we don't have them. Okay. Great. All right, hold on. Scott, I'm going to see. Oh, I can't share your screen for you. Scott, let me figure this out. I'm going to put you in the back. Is that okay?
Sure.
Will we try to work this out? Okay. Let's skip Scott's presentation for the time being and get John Summers to do an update of cancer briefs.
Hey everyone, it's nice to be here. Let me know when you guys have my slides up.
Hey Frank, can we get Jonathan's slides up?
All right. Hi everyone, I'm Jonathan Summers. So the Cancer Brief Project was a HOPE-funded pilot study that we started, a long time ago. It consists of 3 episodes and is looking to test, can videos be used to increase public awareness of cancer clinical trials? Episode 1 was a faux news show looking at immunotherapy. Episode 2 was a promotion of one particular trial and kind of this meta-cinematic commercial. Episode 3 is PSA, public service announcement about DEI and why that's important to clinical trials, and I'd like to share some of our findings. So next slide, please.
Oh, you can.
Oh, that's new to me. All right, cool. So methodology, let's talk about it. So we launched 2 videos. In fact, they're the same videos, 3 minutes and 50 seconds long. Version 1 is, both were in English, but version 1 had Spanish subtitles. Version 2 was English with subtitles. We decided to do this because 85% of videos on Facebook are actually watched without sound. So if you're watching without sound, it doesn't really help if you don't have subtitles. So that was one of the decisions we made. The type of campaign we ran to, it was video views campaign and traffic campaign. We started with video views and switch over to traffic. These are 2 of the basic settings that Facebook uses, and we could use different algorithms to really target that. As Facebook goes, in January of this year, they actually changed how you could target audiences. Previously, we were able to target certain, charities or organizations or hospitals or even metastatic disease. Unfortunately, you cannot do that anymore. It's definitely more broad, and we had to keep that in mind as we thought about how are we going to target this audience and what are we going to go about. So the first thing we did, also from a budget standpoint, we decided to go as large as possible. And Facebook's really good at finding your audience, so let Facebook do that for you. Some of those interests was healthcare, wellness, things like that. The data from our previous episodes showed that people over 45 were our target audiences, so we didn't really go for 22-year-olds, because why? Also, with language settings, in regards of episode 1, we chose people who spoke Spanish, and then we also identified certain cultural things that are popular in the Hispanic or Latino culture, and we listed those, and that would help us find audiences. We also chose cities that had a very large Hispanic/Latino population, and yeah. So next one. So let's go through the numbers. It's launched on September 9th. These were the numbers as of the 16th of October. The total spend, about $1,900. Video 1, $800. Video 2, $1,100. The reach, 148,000 people. And again, it's important to realize when I say reach, let's define our terms, right? So this is when you go to Facebook and you see like a news feed and like this video is for you, opposed to if your friend sends you a funny video of their niece and their dog, that's organic, right? So we have to be, I want to make sure we're laying the terms here. So 148,000 people identified with it. We got 89,429 people to watch the first 3 seconds. We'll get into that, why it's important. Through plays was just about 39,000. Outbound clicks that made it out of Meta, meaning that little, as you'll see in a second, that like click to learn more, we were able to get 1,700 people to click. So what did we do with this information? As info came in to us based on our audience, we were able to figure out certain trends in our users and optimize, as we call, user engagement. So here are some, we identified which thumbnails more people are reacting to and which audiences, as well as headlines. I'll ask Barbara to read this one because my Spanish is terrible, but this was the most popular headline in Spanish. It got the best click rate for that. This isn't surprising, but bilingual people preferred the Spanish headline over English. The thumbnail with a woman had more completed views. Thumbnail with a man actually, though, had more clicks to the DEI page, which is where we were trying to direct them. Now, with this in mind, we were able to target and influence. As you notice, I said it started as a view campaign and then moved to a traffic campaign. So those are two examples how we could leverage the two. The other thing about user engagement, the gold standard in user engagement is when people talk with each other organically. Just think about it, that means they saw it, they had an opinion about it, they understood what it was, and wanted to engage with each other. Out of respect to them, because they didn't know this was from my presentation, I blacked out their names, but you know, some of that is really heartbreaking what you read. And you know, the other one about chemo and radiation, I'm not sure which one tried to kill me quicker. And someone said, didn't take me twice, like, you know, that's the internet for you. And I was really happy to see this organic conversation among people about trials. So let's get into the numbers. Oh, okay, cool. So on top here, we have video 1, Spanish subtitle viewership. Below is the English subtitle viewership. You'll notice in the English subtitle, the reach was about 99,700. In video 1, the Spanish one, the reach was 50,000. But look at the clicks, right? They're almost equal. So what happened? Cool. Look at the clicks, right? 850 to 852. So that's telling us that the audience that speaks Spanish is extremely engaged, right? They want to get right to it. And then if you look at the other statistics in there that kind of jump off is in terms of how many watched, up to 25%. And this is really the inflection point, and I'll get to that in a couple slides later. But you know, in a 3-minute and 50-second video, we're talking about a minute. That's a lot of time. You know, if you think about how long you watch a video for, asking a minute of someone, of a stranger, is a big ask. But nonetheless, there is a substantial, it's 4.57 opposed to the English subtitle, which was just 2.25. So it's telling us a lot about the audience here. And yeah, so we'll move on to the next one. So I want to talk a little bit about back-end analytics for a second. You know, I think this will be something useful. I think Allison mentioned, like, how do we know where people came from, right? So when you have a video, whether you're aware of this or not, and you click on it, it's called a UTM tracking, right? And it knows exactly how you came to that site. So when we went in the back end of Hope Foundation site on their DII page, we were kind of surprised that why is there only 32 clicks? Like, why is there only 32? It does not, you know, make sense. But then when we looked around their website on their various pages, we were able to see 642 new visitors that came directly from our ad campaign. And there are several campaigns. I think all in all, we probably ran 12 to 14 different headlines, different versions, different audiences. And you know, that's great. That's 11.59% increase of new visitors, not just new visitors, of visitors on the whole Hope Foundation site. But it still doesn't track to that 1,702. And the reason is Google versus Facebook analytics, right? And it kind of gets into how the two are designed. I don't want to go like too deep in this, but first reason is ad blockers. If someone is using an ad blocker where cookies are not enabled, there's no way to track it. Google can't do that. So we need to keep that in mind should we move forward and get more granular with this, is making sure that the site that they're directed to has a different way than perhaps Google Analytics to track. The other thing that I would suggest, it's called Facebook, they call them a pixel. You know, it's currently not on Hope Foundation site, and I recommend it because it's a code, and you place it on your back end, and we could do it for you. And it collects data. It tracks not just the data of who came, but where they came from. And then that allows the person to customize ads for that person, right? We already showed that they're interested. Let's keep their interest going and let's tailor something for that. Specifically, if you're thinking about donations to the Hope Foundation page, if you were to have the pixel on the back end of donate, you would then be able to get a good audience, an idea of who are the people that are donating to us and how can we target those people. It was something that would have been, you know, in kind of hindsight, looking at it, that it would have helped more data collection, but we were able to get through that. A couple of the other reasons that the numbers don't exactly match is the mobile cross-device conversion. When you go from mobile to, let's say, your desktop, there's no really way to track that. And considering 98% of Facebook is used on mobile, I think we really need to target the use, not just the use, but the back-end analytics for mobile use. You know, if people used to use desktop to get there, but now they're using mobile, I think we need to update that. The last one is called the UTMs. We kind of talked about this, but there's certain parameters, and 40% of Facebook uses what's called the HTTPS opposed to HTTP. It's just how they got there. But that's meaning that 40% of clicks are going underrepresented. And right now, Facebook is still the largest social media platform in the world, significantly. Though TikTok, Twitter, it's all growing, Facebook is still the big one, which is, you know, it is what it is. So if this is how people are finding the sites, I would really recommend that we build the analytics on the back end that works with that. So let's talk about retention and viewers here for a second. So I kind of charted right here viewership. So let's episode 3 viewership, as you could see, we started off there, you know, starts at 100. That's when someone just starts. Within 3 seconds, it dropped to 59.65. That's actually pretty, oh, sorry, keep forgetting that. Yeah, 59.65. That's where we started. And by 3 seconds, it drops all the way down. And you may be thinking like, what? This sounds terrible. Actually, it's not at all. The average time that someone watches a Facebook video is 4.5 seconds. And the average amount of time that someone even makes it to a minute is less than 5%. But what we noticed that was really interesting that I want to focus on is that inflection point of 1 minute at 25%. So once we go from there, it goes way up if we start looking at our retention there, right? So for the English subtitle video, we started at 2.25. People who made it to 50% is 45.19%. People who made it to 75 is 67. And people that watched 100% is 70%. And then when we look at the Spanish subtitle video, again, it drops significantly to 4.57, then goes up to 54.30, 61%, and then 56%. So what does that tell us? It tells us that we have an incredibly engaged audience. It's telling us that the public does want to know. It is saying that we did a good job with identifying audiences, but we expected that drop right there. And in fact, what I found to be interesting is when you look at all 3 episodes, the yellow is episode 1, blue is episode 2, and then 3, it all follows kind of the same pattern. And, you know, I would have to add up actually the numbers are on the next slide combined. So here are all 3 numbers combined and kind of overviewing. Again, keep in mind, episodes 1 and 2, we targeted specific interests. So episode 1 was immunotherapy, NIH, things related to that. We also had our SWOG colleague, Dr. Sandip Patel. So UC San Diego was one of the interests. Episode 2 was metastatic breast cancer. So we targeted interest groups related to that. Episode 3, again, these are the numbers as the 17th. The reach 150,000. Yeah, so these are our numbers on it. And, you know, hypothetical next steps and conclusion, is media an effective tool to increase public awareness? We believe the answer is yes, very much so. I know Don and I, when we kind of sought this out, it was interesting to look at. I think it would definitely warrant further investigation. But how would that investigation really look? And these are kind of our suggestions. We have enough data from all 3 campaigns to tell you which audiences this plays well with and which ones don't. So the first thing we would do is build lookalike audiences. So what that means is if it played well with an audience of 45 to 52 that were interested in XYZ, we're going to build that audience and go after them again, but in a different way. Another thing is to really look at that inflection point of who made it to 25% and why, you know, they clicked away. for whatever reason, they were no longer interested. And tell me if this has ever happened to you, because it's happened to me. Have you ever watched a video and then stopped for whatever reason, and then later you see the same video again and you realize, oh, I stopped at the same time, right? There's something about that, why is it we kind of lose interest? I think creatively going forward, what made this campaign more successful, it started with a question. What do you think about cancer? Like anyone could have a thought on that. Next, opposed to episode 1, I believe it started with, next, what is immunotherapy, or no, we're gonna tell you about immunotherapy. So it's directing you, it's telling you this is what it's gonna be. The other one was telling you about a breast cancer trial. But in this one, it was regular people and oncologists talking honestly about their experience, opposed to things being scripted and very glossy. So I think that kind of honesty plays very well in this demographic. The other thing is definitely shorter content. As we were saying, kind of the average time they recommend ideally in videos, I mean, you could argue either or, but like 15 to 20 seconds. And that's kind of pushing it. We kind of talked about this earlier, is reconsider the call to action based on the back-end analytics so we could see it. I think that if we were going to do this, we would like to close the circle in that we could do the promotion and then we could also see how it's playing. Because we were really on this end doing the promotion, only later it was like, well, you could see the analytics. I think if we could bring it together, we could do more pivoting quickly and build off those audiences. And lastly, utilize Facebook Pixel. I think it would be extremely useful for these projects, but also for fundraising opportunities. And with that, I will open up for any questions.
So I would love Jonathan to write this up. I think it's fascinating. I don't know what that would look like, but if you're interested in a deeper dive of this project, I think this is fantastic. And I just want to shout out to the Hope Foundation for supporting this work. Please let Frank know if you want to do, I'll have Jonathan sort of lead a group debrief, and then we can discuss next steps outside of this meeting. Is that okay with everybody? Okay,
Please,
great.
I'm not
John,
a data
congratulations.
scientist. Oh, thanks.
Congratulations, this is fantastic.
But I would love your help, so.
Great. All right, so we have half an hour.
Oh, any questions? Sorry.
Perfect. Thank you. Thank you, Jonathan. Thank you. All right, let's go back to Scott Mordler to do a presentation, and then we have one more, and then I think we'll be ending on time.
Great. All right, sorry about that, everyone, but I see it's working, so thank you. So my name is Scott Mordler. I'm a pediatric oncologist out in New Jersey. And this project came about from the initial Cosmo meeting, and we looked to better understand how social media, professional social media use is impacting the emotional health and burnout of oncology professionals. And this project was a collaboration between both SWOG and the Children's Oncology Group. Next slide, please. And I just want to give a quick shout out, went too fast, to my collaborators on this project, Tiffany Lucas, Molly Berry, Liza McDonald, Ariela Marshall, and of course, Don Sun. So, we all here know the importance of social media and how it has benefited our patients. And with individual cancer patients, their support groups, online presence, ability to understand information and education, disseminate education, clinical trial information. But we don't really know how it's impacting or how it's being utilized by our oncology professionals. So again, we were really trying to evaluate the professional purposes and how it's impacting our well-being as professionals. Next slide. So we set out to develop a survey, which was an electronic survey, and it was piloted both by pediatric and adult oncologists. It was then approved by both SWOG as well as the Children's Oncology Group for distribution. And the survey itself included demographics, information about social media use, and then emotional health assessments. So the assessments that we utilized were the 2-item burnout questionnaire, as well as the patient health questionnaire for looking at psychological distress, as well as additional questions about career and job satisfaction, sense of community that social media provides, and imposter syndrome. Then that survey was distributed via Qualtrics to all of the SWOG and COG members, and reminder emails were sent out. Next slide. So we had about just over 3,000 participants initially agreed to participate. We lost 128 people with the lack of demographics. And then the split between that 3,000, 2,127 do not use social media professionally, whereas only 873 were using social media professionally. And then we lost a few more in terms of not full responders. Next slide. So in terms of demographics, these are kind of the averages or the majorities. So in terms of who's using social media, majority were female, that's 74%. The biggest group were those who were born between 1976 and 1984. The majority, about 60% total of social media users were born 1965 to 1984. Almost half were attendings, about 42%. And then we're breaking it down. Then in academics, the majority were in the academic field as opposed to a community practice or private practice. And then we had a large amount of responders coming from the pediatric realm as opposed to adults, and then the other would be other surgical or research fields. Next slide. So what did we find? So in terms of burnout itself, we found that there was a slight increase proportion in the number of responders who utilized social media professionally who were meeting criteria for high levels of burnout. So about 37% were burnt out in the social media group as opposed to 32% in the non-social media group. And then when we looked at overall psychological distress, it was pretty similar. The means were pretty similar, but we started to drill down in terms of the degree of psychological distress. What you can see in that light green towards the top is that there was less severe distress within the social media group. So about 3% in the social media group compared to about 5% in the non-social media group. Next slide. And when we started asking questions about other areas in terms of their work-life integration and their jobs, it was about the same between the social media and non-social media groups in terms of whether or not they would choose that same career or job again and their overall career satisfaction. However, as you can see in the middle graph there, in terms of those sort of turquoise and light green, that those who are utilizing social media professionally reported less good numbers in terms of this was a Likert scale of agree to disagree, and so less of them were agreeing or strongly agreeing that they had enough time for their personal family life. Seems to be a difference in terms of the social media group in terms of their work-life integration. Next slide, please. We then also, oh, sorry that this is not displaying perfectly on your screen, but we asked about the impact that they felt that social media was having on their wellness and burnout. So when asking the social, this is just those who said that they use social media professionally. When we asked them about how did they feel social media was impacting their wellness, as you can see from on your screen, I think it looks kind of that big white chunk was neither positive nor negative, but within the pinkish orange compared to the purple, there were more people who felt that social media was having a positive impact on their wellness. Similarly, when asking them about their social media's impact on burnout, majority, about pretty much half of them said that social media use has no impact on their burnout, compared to when you take that out, there were more people, about almost 20% who said that social media does help to alleviate their feelings of burnout. So there is some discrepancy here between what we found when we specifically asked them about their views on their social media use on their wellness and burnout compared to their general wellness scores. And we'll talk about that a little bit in a minute. Next slide. When we asked about, oh, again, I'm sorry, this isn't displaying nicely, but asking about impact on career satisfaction, there really was kind of positive or no impact in terms of career satisfaction. So 63% reported that social media use does not impact their career satisfaction. But when you're looking at positive versus negative, 27% said it actually does help them with their career satisfaction, as opposed to only 10% said that it worsens their career satisfaction. Next slide. And then when asking about imposter syndrome, because that is something that comes up a lot in social media, and actually very few people responded that imposter syndrome is very prevalent in their life in terms of their social media use. So really very few, majority of them said never, only a few times a year, which really is not indicative of imposter syndrome. Next slide. Then we asked them about, in terms of those positive and negative impacts, what is really affecting their well-being? So when asking about positive effects of social media, things like networking, sharing, recognizing shared experiences, those are really the top features of social media that help improve respondents' wellness. And when asked about negative factors, things like professional anxiety, or the amount of time that they spend online, keeping up to maintain an online presence, those were the most that were frequently noted in terms of negative impacts. And I'll share with you just a few, some of the free text responses that people wrote in when asking about those impacts. So in terms of the positive ones, they said that they felt that it helps with their visibility, which otherwise would be difficult to achieve, that sometimes they would feel less alone, they feel part of a community, that it inspires them, social media inspires them with their own ideas to contribute, and allowed them to connect with others in a time of isolation. The negative responses and qualitative responses that people had are a little bit more stronger wording. So people talked a lot about this false illusion of a perfect life or a false community that's being shared on social media, that that they see other people in similar roles having time to engage in associations, sit on committees, and how are they able to do these things outside of work when all they want to do is collapse on the couch at the end of the day? And then even further, people commented that social media is either toxic or a cesspool or an echo chamber because of politics. So there are definitely a lot of people that were not thrilled with their social media presence or what they're reading online. And then another really major piece that came out of the qualitative analysis was that many people responded or explained that yes, they might have felt that they are burnt out or have decreased wellness right now, but that's not related to social media. That is directly related to other factors like COVID and the pandemic, which I think is an interesting spin and lens to take this data that we're presenting. Next slide. And then lastly, we also asked about the sense of community, and nearly half of them reported that they do feel like they get a sense of community from their professional social media engagement. Next slide. So what were some of the limitations to this study? And you can advance through. So, I think the big one was COVID, like I was just saying. We definitely recognized that there was a lower participation rate within the adult providers. So, thinking about future directions, that's something that we would need to, a barrier that we need to overcome. Survey fatigue, which is definitely real. And then bias from the non-responders. So we know that not all the adults responded, that there's a large network within SWOG, but we really didn't get to a lot of them. So that bias could have kind of gone either way in terms of how that is skewing our data. Next slide. So some conclusions from this project that we've recognized so far is that while yes, social media users did report a higher degree of burnout, they had less severe psychological distress. That social media users reported less work-life integration compared to non-social media users. but they overall had similar degrees of career satisfaction and willingness to choose the same job or career again. We still don't know exactly how COVID played a role in people's emotional health. We can make some assumptions based on other data that has been presented, but it still is biasing these results. And then, furthermore, when you think about, when we asked about people's overall burnout or wellness, but then when asking them specifically about the role that social media engagement plays in their wellness and burnout, that's when people actually reported more of a positive spin or a positive impact on wellness or alleviating their burnout. So next slide. So, next future directions for this, we're still finishing some of the multivariate analyses right now in terms of trying to look at certain demographic factors in terms of whether gender, academics, pediatrics versus adult, type of role that they play in the hospital, and how that impacts their wellness and social media use. And then big picture, trying to think about how to take this data and where to go next with it. We really felt that this was sort of first trying to take the temperature of people's wellness in terms of their social media engagement, and trying to ask a lot about a lot of different types of aspects of their wellness in terms of imposter syndrome, sense of community, career satisfaction. So now that we have this data, really thinking about where we're seeing these signals and what kind of interventions that we might be able to develop in the future. And that's all that I have to share, but I'd love to hear from the audience if they have specific comments about the data or ideas about interventions and the role that we can play in trying to help people with utilizing their social media engagement.
Scott, thank you so much for the presentation. And again, this is an ongoing analysis, but a very successful collaboration of a survey between SWOG and COG. So for that, I think our Dana, who really led the implementation of this, is to be congratulated about it. So you'll hear more about this as we finish the analyses. If you have any questions, please direct them towards Scott. We're really trying to make sense of the data in a way that is not biased. And so giving us your thoughts about what you think the data says is going to be incredibly relevant as we build a discussion. Okay, I'm going to have Dr. Tamimari come up to do our final presentation for this meeting. And she's going to talk about a mobile intervention in the context of CPC, cancer prevention and control.
Thank you so much. I promise I won't keep you long. Just a brief outline of what will be my brief presentation today. Just giving you the context of what are the mHealth tools, particularly the ones that my research team is using for tobacco prevention and cessation. And then the two flagship studies that we have currently and where we are recruiting participants. So within the realm of the information technology, digital technologies, health information technology, when we start using Epic and all these platforms to help our day-to-day activity. And then it came pretty much in the past 2 decades, the e-health platforms helping to many of us to conduct research. But what was really a game changer is being the smartphone that is allowing us to access multiple smartphone applications, and is giving us the capability to have a portable tool, a mobile tool, and there's where this term of mHealth tools coming from. So for the past 15 years, I've been using mHealth tools to sustain my research agenda. And the first study that I'm going to present is a study funded by the NCI. It's a 3-year study where we are offering a smoking cessation text-based program for LGBTQ+ smokers. This is well, it is known to many of us, particularly in the field of tobacco research and nicotine addiction, that the sexual and gender minority groups has been viciously targeted by the tobacco industry. And as a result, well, we have this high tobacco use prevalence. This is a study that I conducted with my team back in 2014. We went to the Houston Pride Festival and Parade and collected data. And just to give you a little bit of context, we found 55% of those responding our survey being current smokers. Just to give you some perspective, at that time, the general population was smoking 17%. So that's huge. That is a tsunami of tobacco-related conditions, including cancer, that sexual and gender minority groups are going to face and are facing currently. That without counting that there are many other tobacco products, smokeless tobacco, hookah, and now, of course, electronic cigarette that is being a boom, particularly among youth. So based on this idea is where we came with smoke-free SGM, SGM standing for sexual and gender minorities. And what we propose is to test the feasibility of a text-based intervention for smoking cessation, specifically tailored for sexual and gender minorities. And where are we right now with the study? Well, we already developed the text message library, adapting it to fulfill the needs of the LGBTQ plus smoker. And that is responding to minority stress due to stigma, discrimination, and many other social, personal, family factors that are affecting or increasing the stress level, anxiety, depression among this population, and they're using smoking as a coping mechanism, as one of the coping mechanisms. Then we pre-tested the text-based platform in the phase 2. We did that early this year. And that pre-testing phase included the whole machinery of mailing nicotine patches, having the screening and the consenting gone through video conferencing because we were in the COVID time, so everything needed to happen virtually. And right now we are ready to roll up. Actually, just last week, we started recruiting for the pilot randomized controlled trial. And this is just highlighting a few of the things that we tailor in the text messaging platform. We have a quitting coach. We name it Alex, so it's a non-binary name. In that way, if the person is having the cell phone sitting on the sofa and they are not out to their family or friends, it is not a messaging coming from, oh, this is the LGBTQ smoking cessation program. It's just Alex sending a text message. And then we create a specific text messages for tackling minority stress among this population. And the beauty of this program is that it's not a passive one where they just keep receiving messages. These could be bi-directional messages. So there are key words that if they're in a stress situation, like, oh, I'm ready to come out and I feel like I need a cigarette, or I'm meeting with some friends, if they text stress, then they will get personalized text messages back where we gave some strategies for them to avoid any lapse. And this is the machinery that involves the entire project that we tested in phase 2, having the screening via telehealth, the signing of the informed consent electronically, mailing to the address of these individuals the nicotine patches, having a saliva test to biologically confirm a smoking status. And now that everything is sound and ready, we are ready to roll out, and this is pretty much the design. It's a 2-arm randomized controlled trial where one arm of 40 individuals is going to receive a text messaging smoking cessation program, but it's the one developed by the NCI for the general population, and the other 40 people will receive the tailored version in an attempt to prove our alternative hypothesis that if you tailor a program to the intended audience, the retention, the engagement, it will be higher. And we are going to follow them at 3 months and at 6 months. So these are some of the metrics that we are going to assess: recruitment rate, retention rate, engagement with the program, smoking abstinence rates, of course, and then we will have an individual interviewed at the end just to assess usability and acceptability of the program. This is the flyer. So if you have among your patients, and we are talking about individuals who self-identified as sexual and gender minority group, any in the spectrum of this population, please refer to our team. There is a QR code. We are rolling this study all over the United States. At this moment, we only have it in English. We are working now into a new submission because we're going to expand it to a Spanish population. Being myself a Latina, then of course a huge bias in that intention. And then there is decision T. So I present an mHealth tool for the patient. And imagine a cancer patient self-identified as LGBT individual who needs support in their quitting effort. Now I'm going to switch to the other side of this spectrum, the provider. One of the problems is that people think that for offering smoking cessation counseling, you need to, well, ideally, you should have a smoking cessation training, but not necessarily. Any healthcare provider, and by that I am meaning not just physicians, non-physicians healthcare providers can give this service to patients. And as you know, many of the cancer patients really need, for having comprehensive cancer care, smoking cessation services, because some of the cancers are related to smoking. So what we did is we developed a smartphone application. We call it Decision T. And the story behind this is the Hope Foundation Impact Award. I am an awardee of this funding mechanism, which was intended for a training program in Colombia, Peru, and Brazil. And we successfully conducted the training program. We trained 90 cancer care providers in smoking cessation in Latin America, in these 3 specific countries. But unfortunately, COVID hit when we were training the last group in Brazil. So what we did, we used some of the funds to develop a smartphone application, because the trainees were complaining that, oh, we need to go back to the slides to make sure what are the patches, what is the dose, how we need to do this and that. And it's how the idea of creating a decision-making algorithm for the provider that can walk them through the process of giving a smoking cessation service. So we call it Decision T because of decision-making process, and T because of tobacco. And we already tested with 18 or pre-tested with 18 HIV care providers successfully. In less than 4 minutes, they were able to walk through the 5 A's model, which is the typical model for a smoking cessation, when you need to ask the patient, advise the patient, arrange, and you go through the 5 A's, and the algorithm help you with that. Not only with the counseling, because we embedded also messages for taking the feedback of the trainees, like, I don't know what to say to a patient who is dying. And it's like, why do I need to quit smoking? So we embedded certain sentences that can help them to initiate that conversation with the patient. And this is kind of how it looks, the smartphone application. I won't bother you. I can give you access if you would like to see it. I have it in my phone. And that's another effort that we are going to expand to Spanish speaking healthcare providers. And we're working right now with Puerto Rican colleagues to see if we can test it there. But whatever you are choosing in the app is guiding you and popping up with different screens, so you can complete the counseling session all over, from studying if you are a heavy or light smoker to what is the dose of nicotine patches that you will need. And what I need now is, if any of you knows healthcare providers, cancer care providers who have an Android, who would be interested to test it. You don't need the patient. We have in my research team people who will fake to be a patient. And in a telehealth session, 3 mock sessions, we will give you access to the app, you will test it, and you will give us your opinion to see if we are on the right track. And this is the QR code. This is the email address of my research team. It's the Health Equality Research Group. And if you are interested, just reach out. And here is my, finally, I am also part of R25 training program, where we are training scientists and clinicians on cancer care for sexual and gender minority individuals. Everything from epidemiology, community-based participatory research, behavioral interventions, clinical aspect of cancer research with LGBT or for LGBTQ+ population. We are opening the application period that will close next month. And it's fantastic. We tested last year, NCI we finalized the curriculum development, and we were given a 5-year grant for having 5 cohorts. The one next year will be virtual, but then the other 4 will be one in Houston, the other one in New York, the other one in Florida, and the last one in New Mexico. So thank you so much with this information. I hope that we can continue collaborating. I want to wrap up just saying thank you to the Help Foundation, who have, this organization has been key for my career development. Thank you so much.
All right, that's great work. Thank you very much for presenting it. And again, for the thoracic liaisons, if you're here, please mention this as part of your updates from the Digital Engagement Committee. Thank you very much for everybody for joining. I will be remaining a member of the committee. And thank you very much for everybody for your help in making this a success at SWOG. Thank you.